And welcome, sir. Let's begin the session.
Brilliant. Afternoon, everyone. Venki, it's an absolute pleasure and honour to be interviewing you. I don't want to come across as a science fanboy, but we'll have to wait for people to come up to hug you at the end of the session once we've finished. I think, actually, for the purposes of time, we're going to cut straight to it. So we're here to discuss this book, The Gene Machine, which is really rather masterful and actually is 3 books in one. Because not only does it tell us a great deal about the science that underlay Venki's honour of the Nobel Prize in 2009, but it also is 2 other books within the same book. So it tells us a lot about the nature and the process of science, as well as the history of science. And it's also a personal memoir. And I hope that over the course of the next 44 minutes, we'll get to all of these areas. But I just wanted to start with the science, if I may. So you describe the ribosome as thus: virtually every molecule of every cell in every form of life is either made by the ribosome or made by enzymes that are themselves made by the ribosome. The ribosome lies at the very crossroads of life.
Yes.
So please tell us more.
Yeah, so it's interesting. Somebody described the ribosome as the most important molecule that nobody's ever heard of. And one reason is that we all tend to emphasize DNA. So the average person, if you say, what do you know about DNA? They think they know something about it. But actually, they don't really understand DNA either. What they don't understand is that, yes, DNA is concerned with our heredity and so on, but they don't realize that DNA is not just about heredity. It's also about building building an entire animal from a single cell. And what DNA actually has is information on how to make proteins. So most genes, there are some genes that code only for RNA, but most genes basically contain information on how to make a particular protein. Now, again, if you ask the average person on the street, what are proteins? They'll say it's something good to eat. You need it, to be strong, and it's good for muscles and so on, and you need enough of it in your diet. But actually, proteins carry out almost all of the things that we think of as characteristic of life. For example, the reason you can see me is because of a protein in your eye. The reason that you breathe oxygen and can use it is because there's a protein that traps the oxygen in your lungs and takes it into your tissues. Your muscles that make you move are made of protein. Your skin is made of protein. And even memory is formed by proteins. So you need protein synthesis of specific proteins in order to form memories. And antibodies that fight infection are made of proteins. So you get the idea. Proteins carry out a wide variety of things. So the question is, how do you go from genetic information of the sort that's present on DNA to proteins? Well, it turns out DNA and proteins are 2 very different kinds of polymers. DNA is a polymer consisting of 4 types of building blocks called bases or nucleotides. And proteins are made of 20 or more types of building blocks called amino acids. And their chemical nature is very different. So you're going from the language of proteins, think of it as a sentence written in a 4-letter alphabet, to the language of proteins, which is a different language with a 20-letter alphabet. And interestingly, biologists call this process translation, because you're going from one sort of language to another. And the particle that makes it all happen is an enormous molecular complex called the ribosome. And it was discovered in the 50s and late 50s. And then gradually people figured out how it reads genetic information to make proteins. And so that's really, in a nutshell, what the ribosome is.
So this is the machinery through which all of life is translated, is that right?
Right. And in fact, it probably predates DNA. People think that life began with a world which primarily consisted of RNA with maybe some small proteins. And then it was the ribosome, which is made of RNA itself, that it evolved and started making proteins. And gradually, this information for making proteins became encoded. And then that really was the start of genes on DNA. So there is this idea that DNA actually is a latecomer, and the ribosome is actually older than DNA.
So, one of the things that strikes me is that obviously the structure of DNA was solved in the early 50s, and you won your Nobel Prize in 2009. Why, can you explain why there was such a delay between understanding the structure of two intimately associated molecules?
Yeah, so in fact, the ribosome was discovered not long after the structure of DNA. It was discovered in the late 50s, and the DNA structure was solved in 1953. So only a few years later, people had already identified the ribosome as where proteins are made. But DNA is a fairly simple structure. It was a remarkably important structure because the structure itself told you, provided really strong hints of how DNA would carry information. So DNA, for example, consists of just two strands woven around each other to form a double helix. So it's a very simple structure, and the relationship between the two strands, where each base recognizes a complementary base on the other strand, also provided biologists very strong hints about how DNA would replicate and how it might carry information to make proteins. Now, the ribosome, on the other hand, is enormously complex. In humans, it's about 500,000 atoms. And in bacteria, it's about half that. But it's still a lot of atoms. And it doesn't have that neat symmetry of a double helix. If you look at a picture of a ribosome structure, it looks like an enormous bowl of spaghetti with a lot of vegetables thrown in between, the vegetables being the proteins, and the spaghetti being the RNA that makes up the ribosome. So it's a highly complicated structure. And in the 50s, Jim Watson, whose lab pioneered work on the ribosome, once introduced me at a talk, and he said, when he realized how complicated the ribosome was, he realized they would never know its structure. And he was right. In the 50s, there was the technology to determine an atomic structure of the ribosome, so you could visualize the ribosome in atomic detail, didn't exist. And for a long time, until recently, the only technique to reveal that sort of structure was a technique called X-ray crystallography. And what this requires you is to make crystals of the object. Now, that's itself complicated because it requires you to form regular three-dimensional arrays of something large and floppy like the ribosome. And that didn't happen until 1980. And then you required very powerful sources of X-rays, which were synchrotrons, which again started in the 80s. And then you also required much more powerful computers and computer graphics and the software to do all the analysis. So for a variety of reasons, it just simply took a very long time to determine its structure. I should point out there's now a technique called cryo-electron microscopy. So today, a structure of a ribosome would take an undergraduate project student about 2 weeks to solve. So that's how science has moved on, and it's probably lucky for me that we solved the structure before cryo-EM came along.
Very lucky indeed. So I wanted to just reflect back on the book because you gave us a very quick summary of some of the challenges involved in some of the work that you did. In the book, you very clearly demonstrate the fact that science does not move in a linear way, that there are sudden changes in terms of our understanding or the technology that's available. And you also very clearly illustrate that by and large, science is a communal, collaborative experience. But I just want to take a little sidestep into the personality of science, and in particular, the personality of scientists. So one of the things that struck me was basically the first quote in your book is from Richard Dawkins. I'm sure many of you will have heard of Richard Dawkins, who's a very famous evolutionary geneticist in Oxford, who describes Venki as a sort of nice Jim Watson. Which is a compliment to Venki, but it's not necessarily a compliment to Jim Watson. And in other bits of the book, Venki also describes some fairly unpleasant characters, some of whom wrote rather snotty letters to you at times. And what I wanted to ask you specifically is how important do you think personality is in the world of science, in what should be a communal endeavor?
Well, you know, we think of science as this noble quest for the truth done by people who are very objective and altruistic.
Is it not? Is that not the case?
And no, obviously not. Science is done by humans. So scientists showed the entire spectrum of human behavior, from greed and ambition and jealousy, also hard work, persistence, drive, but also altruism and kindness. So you get the whole range of human behavior within science. So science is really a microcosm of the larger society. And yes, scientists will collaborate when it's in their interest to do so, just as businesses will. For example, Apple and Samsung are extremely competitive about the mobile phone business, but they will collaborate. For example, Samsung will make chips or glass that's required and so on. So the same people, scientists may collaborate on one thing while competing on another. So it's not really, I don't see it as any different from anywhere else.
So talking about this duality of competition and collaboration, you know, one of the people who features very strongly in your book is your fellow Nobel Prize winner, Ada Yonath, who was, you know, at certain points your nemesis. She was essentially your Lex Luthor to your Superman at certain points. And it's very clear that the nature of your relationship with her was rather complicated in scientific terms.
Yeah, although I would say I was on much better terms with her than Tom Stites was. I mean, they really went at it hammer and tongs. But I think part of the problem was that Ada Yonath had obtained the first crystals of one of the subunits of the ribosome in 1980 when she was working with Heinz Gunther Wittmann, who was the director of the Max Planck Institute in Berlin. But then, if you fast forwarded 15 years, there was still no real progress towards an actual X-ray structure. And after a couple of talks in 1995 and 96, a number of us decided that, well, maybe, you know, I mean, to put it bluntly, some of us felt she wasn't up to it. She had never solved a large structure before of any kind. And we thought the field could use both new insights and new approaches and also some competition. Now, the problem was that she had had this field so long to herself that she didn't take very kindly to the idea that suddenly 3 other groups were entering a field that she thought was hers. And so there's a lot of tension. And of course, there's a lot of scientific disagreements as well. I mean, a number of us didn't really, you know, agree with her on various aspects of her science. So it led to a lot of acrimony. But I think in the end, I mean, my view is that she did deserve the Nobel Prize because she got the first crystals and sort of set the agenda, if you like. And so in a way, I felt that was a very important contribution. Now, in terms of my personal relations, I don't know what they're like now, but I will tell you a very funny story about my book, Gene Machine. So, you know, the Royal Society was very nervous about my book. because I was the president at the time. And so they wanted me to give her a copy. And that copy was returned to me and, marked return to sender. And then she wrote to me through a friend and said, well, I understand you've sent me a book, but I never received it. So I sent it to her again. I told her, look, it came back. It was postmarked with a Weizmann Israeli postmark, so it clearly got to Israel. So anyway, she then read the book, and I didn't hear much from her. Then I met her about a year later for the first time in person since the book came out. And it was at a meeting in China, and I met her at the end of the meeting because I'd only arrived on the last day. And so I just said hello to her, and I wanted to quickly say goodbye and sort of leave. And she said, no, wait, I need to talk to you. And I thought, oh my God, she's going to have a list of complaints about the book. But then she said, I was in Mexico giving a talk, and afterwards a young fellow came and wanted my autograph. And I said, yes. And she said, you know what he did? He reached into his bag and pulled out a copy of your book and wanted me to autograph it. And I just sort of laughed, and I said, well, did you? She said, no, of course not. I told him I hadn't written this book, and I had nothing to do with it, so I wasn't going to autograph it. So I said, oh, and she said, do you know what he said? He said, but Professor Yonat, there's a lot about you in this book. And so at that point, I burst out laughing. And she said, I knew you'd find it funny, so that's why I told it to you. So then I thought, well, actually, she sort of accepted it and moved on.
So you made your peace in some way.
I guess so, although, I don't know. I don't think we'll ever agree on relative contributions and the science and so on. I think that'll be left for other people to decide.
So I think you're getting a bit of a flavour of this book, Gene Machine. It's not only about the science, but it's also about the drama, almost the soap opera, if you'll forgive the expression, of the race for the secrets of the ribosome. One of the things that strikes me is you provide a great deal of detail about the extreme highs of research, but also the terrible lows, the points at which you thought that nothing was going to work, your career was going to go down the pan, you were never going to get another job again. Where do you think that quality that kept you going came from? Was it optimism? Was it curiosity? Or just relentlessness?
I think, I had several setbacks. First of all, I didn't go to a top, I got into some top universities in India because I was a science talent scholar, but I was only 15. And, my parents didn't want me to go off to Delhi University or something. And so I went to my local university in Baroda, which had a very good program. But then I was impatient to go to the US. And so at the age of 19, I started graduate school in physics at a university in Ohio. But by the end of that, I like to think that by the end of my 5 years, the only notable thing I accomplished was I met and married my wife. But, because my physics was completely insignificant. And so I thought, well, what do I do now? I could have stayed on in physics and gotten some sort of teaching job and maybe done some very boring physics. But then I thought, well, maybe it's time to switch fields. So I went back to graduate school, which was very unusual in that, you're going back 5 years, and I actually took undergraduate courses. So I think that the whole thing was really to keep giving myself second chances. And then I ended up working using a not very useful technique in biology, a physical technique called neutron scattering, which is of very limited use in biology. And so there too, I thought, well, this is not going anywhere. And I thought, well, I should go away on sabbatical and learn crystallography. And I actually went to Cambridge, which is where I eventually ended up many years later. And so I think it's all about giving yourself second chances, not so much. I don't think there was more to it than that.
So you think it was purely the case that you gave yourself second, third, fourth, fifth, and sixth chances?
If something doesn't work out, you sort of try a different path or either a different field or a different technique.
I mean, in a way, it's
or also moving. Sometimes you find the institution is limiting, and you need to go somewhere else to do better work.
I mean, in a way, it's a story of great optimism, because for all those people who are in the midst of their degrees or PhDs who think they're failing, they may go on to win a Nobel Prize.
That's true, but I like to think most scientists are actually optimists, because when you do science, most of the time nothing's working. And the reason is that as soon as something is working, you very quickly do a little body of work and then you publish. Then you move on to the next stage or the next problem, and again, nothing is working. So in terms of the time spent, most of the time nothing's working. And so I think scientists are basically optimists, and those who are not optimists simply leave science because they can't take the constant struggle, because it's not like turning a crank and getting results all the time. That's not actually science.
So you've obviously spent your entire working life in science, and you've mentored huge numbers of people. You've had lots of people pass through your various labs. Are there individuals that you see and you think they have got the qualities required in order to be the next president of the Royal Society? And if so, what are those qualities? Or is it just a matter of luck? I think you alluded a little bit to this in your talk yesterday.
Well, I don't know about the president of the Royal Society. I'm astonished that they elected me in the first place. But anyway, but I think in order to be a leadership position in science, there are several things you have to have. One is you have to be a highly credible scientist yourself. That is, you have to be respected in your field and preferably in the broader science community, because otherwise when you make difficult decisions or statements, you won't be able to carry the community with you. They have to trust that you are, top-notch or highly respected and a person of integrity. The other is that you have to at some level, you have to like people and the community. You can't be, there are many really brilliant scientists who never want to be president of the Royal Society, and also they wouldn't be even asked because they're often highly introverted, very narrowly focused on their field. They're brilliant in their field, but it's not necessarily what would be good for a leadership position. And I think a third thing, apart from liking people, is some concern over broader issues, like where does science fit into society, into government, into international relations, and so on. So it requires a certain combination of those, I would say.
So I wanted to come back to that role in a minute, because one of the other things that you're very consistent about in your book is describing yourself as an outsider. And I just wondered if you could explain why it is that you felt an outsider.
Well, I think that the feeling of an outsider doesn't have to do with any reality. It's a perception. So, for example, I was born in Tamil Nadu, and only spoke Tamil until the age of 3. And then I moved to Gujarat, to Baroda. And in fact, the first memory I have is of standing in a playground and not understanding a word that the other kids were saying. And so that, when you see that as a child, it immediately gives you that feeling of an outsider. Then when I was 8 and 9, my parents went to Australia. So then again, I was an outsider in Australia, in Adelaide. A great cricket ground, by the way. And then I came back. Then I went to America at the age of 19. Again, I was a sort of outsider. And also, there are different kinds of outsiders. You can be an ethnic or linguistic outsider, but you can also be what I call an educational hierarchy level outsider. So I hadn't gone to Oxford or Cambridge or Harvard or Berkeley. I had gone to Baroda University and to Ohio University. So I was an outsider when, for example, when I went to do a postdoc at Yale. I felt like an imposter, like all these people are very smart and I'm they're going to pretty soon find out that I don't actually know anything. So you get that feeling of being an outsider. And then eventually, of course, I moved to England. I hadn't moved completely de novo because I'd done a year of sabbatical there and I knew the people at the LMB. But still, it was a different country. So rather, although it's Anglo-Saxon like America, it's got somewhat, let's say, different customs. Two countries divided by a common language is one expression. And that also felt like an outsider. And so that feeling sort of stays with you. And although as I became more and more recognized and got various honors and I became more and more part of the British science establishment, but nevertheless, you still feel internally that you're something of an outsider.
And do you think that helped or hindered your career?
I think it's a great, a fellow American expat in the UK who works at the Crick Institute said, Venki, it's not a bad thing to be an outsider. For one thing, if you're an outsider, you don't get pigeonholed into various categories. I mean, if you're born in Britain, you often will get pigeonholed. Are you a northerner? Are you or a southerner? Are you working class? Or did you go to public school? There are all these little subtle pigeonholes that you sort of fit into. If you're an outsider, they don't really know what to make of you, and then they have to treat you as an individual. And I think, and then you also don't have particular prejudices of growing up in that society and so on. So in a way, I became Royal Society president right when Britain voted to leave the EU during Brexit. And being an outsider in that sense actually helped me in talking to some very right-wing anti-EU politicians because they didn't see me as the typical Guardian reading academic from Britain. They saw me as this sort of Indo-American and they took me at face value.
It's really fascinating for me because in my mind, you are at least in scientific terms, the insider's insider, having done what you've done. And which was why it was kind of shocking for me to hear you write in your book some of the things that we as academics all talk about constantly, which is that science in some respects is in a bit of trouble. There are structural issues with science. And I think, if I can quote you, you use terms like cronyism and corruption in certain aspects of the scientific world. I wonder if you could expound on that, particularly given the position that you were in.
Yeah, so I think that there's a problem, and that is people will often think more highly, and this is just not restricted to scientists, it's human nature. They'll think more highly of people that they've known, that they've grown up with, they've networked with, known for a long time. And if somebody's an outsider, they're looked at somewhat more critically. I mean, that's simply a fact of life. And I do think that when it comes to apportioning credit, when it comes to publishing papers and so on, it always helps if you're from a good university. It shouldn't help, and in theory it doesn't matter. But I think human psychology being what it is, it often can matter. And cronyism and stuff, I think there's a lot of, I was talking particularly about awards and rewards in science. When people have done a bit of good work, they start getting prizes. They're sort of low-level prizes, and then they move on. Then they become elected to their academy, and then they get international prizes, and then they eventually get a Nobel Prize. And there's a sort of trajectory. And the problem is that very often what will happen is some jury will decide to give somebody an award, and then subsequent juries, that greatly increases the likelihood that other subsequent juries will also give the same group of people an award instead of thinking, well, are these the right people and should we award some other field and so on. So there's a kind of snowballing effect, and you often find the same people winning lots and lots of awards when in fact there's so many great discoveries and they could spread them around.
So whilst we're on the subject of prizes, I have to rather shamefully ask the question that you must get asked all the time, which is, what did it feel like when you got the phone call to tell you that you had won the Nobel Prize?
So it's a very strange day because I bicycled to work about 5 kilometers and from a small village just west of Cambridge. And so my bicycle developed a flat tire about almost exactly halfway through, so I couldn't go back and fix it, and I ended up walking my bike to the lab. So I came to the lab somewhat late and grumpy, and I want to give you a little background to this. So when you do something really significant, what you often find is that you keep getting invitations to conferences in Sweden. And this happened from 2000 to about 2004. And at the last of these meetings that I attended, a ribosome scientist from Sweden came to me after my talk and started just having a vitriolic fight with me about one aspect of the ribosome. And I found the whole thing unpleasant, and then a mutual colleague kind of separated him away from me. And then a few months later, I found that he had been appointed to the Swedish Nobel Committee for Chemistry. So I thought, well, there's absolutely no way in hell that I'm going to get a Nobel Prize. And also many of the other sort of international prizes for the ribosome, except for one big one, all the others had gone to other people. And so I thought, well, clearly the community might think I did something important, but not sort of one of the 2 or 3 who really mattered. So when this phone call came, I was already grumpy, and I was extremely skeptical. I thought, well, this is, this sounds like a prank. And so then I said, you've got a great Swedish accent, but I'm not sure I believe you. And I heard a huge burst of laughter, and I realized I was probably on a speakerphone at the other end. And then I said, well, if it is true, I want to speak to the scientist. And so sure enough, that guy came on, and he told me, well, Venki, I'm here, and it is true, and I want to congratulate you. But I also want to say this is the last time I'm going to congratulate you. So he was still, ready to pick a fight with me the next time we met.
Yeah, I'm sure you didn't mind too much at that moment in time.
But one thing I did in looking back is I deeply admired the guy's integrity. Because, in these committees, all deliberations are secret. He doesn't even have to say Ramakrishnan is a bad scientist or this work is terrible. He can say, this is great work, but it's not quite the same level as the other 2 or 3. And that would have been enough to sink me, right? But the guy put aside his difference about one area and sort of looked at the bigger picture and decided probably to support me. So I have to say, I greatly admire his integrity. And that, I'm not sure how many people would have had that.
Tell us about what difference the Nobel Prize has made to your academic life.
Yeah, well, one of the people on the Nobel Committee said it's the kiss of death, because what happens is you're doing well in science, and then you get the Nobel Prize, and suddenly you start getting invited to meetings to do this, that, and the other, and sit on committees and so on. And pretty soon you're just distracted. Of course, many Nobel laureates get the prize long after they're active scientists, long past their peak. And they go around swanning all over the world. They become what I call airport scientists. And, that's okay. They enjoy their time in the limelight. So what I tried to do in the first few years anyway was try and restrict the number of engagements that I had and really ask the question, is this something I really want to do? And also, would they have asked me to do this before the Nobel Prize? So that really kept me grounded, and my lab was very productive in the few years after the Nobel. I think what really did make a difference was the Royal Society presidency. And of course, I was very flattered to be asked and you know, I enjoyed doing it. It was very interesting. I wouldn't say enjoyed, but it was a very interesting experience, and I'm glad I did it. But the thing about it is that really took me away from Cambridge and from the lab, and had my mind thinking about other things constantly. And so that I did feel affect my science.
So do you think you've been partially affected by the curse of the Nobel Prize, or do you think you have successfully escaped it?
I think, you know, my lab, I'm going to shut down my lab at the end of the year, but we continue to be active in publishing in top-tier journals and trying to do what we think is the next important question in our field. So I wouldn't say in that sense it's affected me. I still live in the same sort of what the British call an ex-council semi-detached house, and I still ride my bike. It's the same bike, actually, to work and back. So my personal life hasn't changed a huge amount. But of course, I do get lots of invitations that I wouldn't get if I didn't have a Nobel Prize. And also...
Like Kerala Literary Festival?
Yes. I don't know if I would have gotten an invitation if I didn't have the Nobel Prize and had written the same book. Maybe I wouldn't have been even able to publish it. You know, they might have said, "Wow, this guy's a loser. Why are we publishing this book?" So I don't know. So those subtle things, and of course the Royal Society presidency. There's no way in hell they would have elected me as their president if I didn't have a Nobel Prize. You know, they probably wouldn't even have heard of me. So that's just how it is. So there are things that come to you after the prize that you didn't get before.
So before we move away, and I am keen to move away from the Nobel Prize because that's only one aspect of what you've achieved. What I was struck by yesterday in your session with Abraham was the fact that everybody is eager to bask in the reflected glory of the prize, and he described you as a son of India. Whereas, you know, what is very apparent to me is that there is no single country or system that I think can claim responsibility.
No. You know, there's an old saying that success has 1,000 fathers and failure is an orphan. So that's just how it is. And in my case, I would say there are at least 3 countries to whom I should give credit. I grew up in India, and I was given my sort of school education and my undergraduate education here, and with many dedicated teachers. And then I really learned how to do science and learned, you know, higher level science, and then learned about how to do research. And also, it fostered my research for almost 30 years. So that's the US. And then Britain, the MRC Laboratory of Molecular Biology, to which I moved, really gave me the courage and the freedom to tackle big questions. And so I would say all 3 countries have been extremely supportive of me. And so in some sense, I would say the science part actually is a result of the US and Britain. But, you know, you have to have a foundation before you tackle that, and that came from India.
So we've got about 5 minutes before we go to questions, and I just wanted to ask really 2 specific questions. The first is, you described the LMB, the Laboratory for Molecular Biology, which is the lab that you are based in Cambridge, as a mecca. I think that's the title of one of your chapters. What is it that you learned there about the practice of science that allowed that laboratory to be so productive and so influential on the world stage?
Yeah, so the MRC Lab of Molecular Biology is a strange beast because it was founded by people who were not actually biologists. In fact, the term molecular biology didn't even exist and really was propagated partly by the MRC Lab. They were physicists and chemists who had banded together to study important biological questions. And one of the first 2 things that came out of it was the structure of DNA and the first structure of proteins. So 4 people won the Nobel Prize in 1962 for work done right at the start of the lab in 1962. And it had a predecessor in Cambridge which was an MRC unit. Now, I, of course, had heard of the MRC lab, and so when I wanted to do a sabbatical, it seemed the natural place for me because first of all, my wife and I are sort of Anglophiles. I mean, not sort of, we're actually Anglophiles. We liked Monty Python and P.G. Wodehouse and all of these things. And so we thought it'd be great to spend a year in Cambridge. And it's also the lab where protein crystallography began. So I thought, what better place to go and learn crystallography? And I knew it was a very collegial place. But when I went there, a number of things struck me. So my host was actually Aaron Klug, who was the director of the lab. But he was very busy. So he had assigned his colleague, John Finch, to take care of me. Now, John Finch was a fellow of the Royal Society. So when I arrived, he said, I'm sorry, we don't have a desk for you to sit in. It's crowded, and we have to figure out where to put you, and you'll have to just stay in the library for a couple of weeks until we find a spot for you. So I looked at him, and I said, look, I could just take a desk in your lab. And he just sort of smiled at me, and then I realized he didn't have a lab of his own. This is a fellow of the Royal Society. In America, it'd be unthinkable that a National Academy member would just have a little desk and a little bench. But that's what he had, okay? And so then the other thing that struck me was I got in at about 9, and about 10 o'clock or 10:15, he came up to me and said, would you like to go have some coffee? And I said, I'm sorry, I only just got here. And somebody else who was there said, he hasn't understood our ways yet. And so then I realized that what they do is they take breaks at 10, somewhere around 10:30 or 11, just a 15-minute break for coffee. Then they have lunch at 12:30 or 1, maybe half an hour. Then they have tea time at about 4, another 15 minutes or so. And what this does, it turns out nobody can really concentrate well for more than a couple of hours at a time. And so these short breaks actually are rejuvenating, but they have a more important purpose. They built a non-hierarchical canteen. So the lab director sits next to a first-year graduate student or even next to one of the cleaners or an electrician. So there's absolutely no hierarchy in the canteen. There's no faculty club or senior common room or things like that. And so everybody feels part of the same enterprise, and they all talk about science in an informal way, and lots of ideas have come out of these informal discussions. Now, a third thing is that group size at the LMB is deliberately kept small. And the idea is, people who are well known can often amass lots of resources and build up huge labs, 20 or 30 people. The problem is nobody has more than 2 or 3 good ideas at once. And usually, if you have one good idea, you're lucky. And so they end up diffusing their efforts. But if you have a small group, it forces you to ask, what's the big question in my field? What am I really interested in? And focus on that rather than all these secondary questions. And that's why when I went to the LMB, I found almost everybody was working on a really important problem and not just doing the next incremental bit of science. And so that was helpful. And that's possible because the funding is stable. And the funding is also based on a personal site review where you have a chance to explain to the review panel your ideas and your progress. And so it allows you the luxury of taking on harder problems than you might be able to in other environments, like in a university where you have to depend on short-term grants. So I think for a variety of reasons, the LMB model has been very successful, and I'm surprised it hasn't been duplicated more often.
Well, Venki, I wish we could extend this session for another half an hour. I've got so many more questions to ask, but I'm sure the audience have got lots of questions. I just wanted to finish with a quote by one of your favourite famous researchers, Richard Feynman, who said, "Our responsibility is to do what we can, learn what we can, improve the solutions, and pass them on." And it sounds like you've achieved all of that.
Well, I've lucked out, as I say.
Venki, thank you very much. Thank you.
Hello, good evening, and thanks for a wonderful session. I just want to ask you, besides working in the field of science, what is your process of writing?
Oh, that's very interesting. So I think I don't have a fixed process, but it is important. Obviously, you have to do all the obvious things like have a reasonable outline and some structure. The structure doesn't have to be very rigid because it can evolve as you write, but you have to have some overarching structure or you'll just wander around and not actually finish anything. But then you do have to make sure you simply put in a certain number of words every week. And whether you do it all on a weekend or whether you set aside a couple of hours every day is very much a personal thing. I found I would often write on weekends. My wife likes to go what the English call rambling on Sundays, and I am not, except in the summer months, I'm not fond of walking in miles of mud and rain. So I would often write on Sundays, but I would also write on long airplane trips. So I would sometimes I've banged out a chapter on an airplane trip. So I think it varies a lot, but having regular writing is essential. Otherwise, the ability to restart the process is a real barrier, I think.
Venki, I just wanted to ask you, so one of the issues that I struggle with is that as scientists we write in a very impersonal, anonymous way. And what's very clear about this book is it's written in a very personal way. Yeah. And so how easy did you find it to make that sea change in terms of your approach to writing?
I didn't think it was very hard. I have a confession. So when I was a physicist, I thought biology was boring, a bunch of facts that you had to memorize and not much, thinking behind it and so on. And the book that changed my mind was actually The Double Helix. And I know the double helix is extremely controversial and so on. You all know about it. And also Jim Watson has not exactly endeared himself to the larger community, including Indians, by the way. He's had his say about Indians as well. But nevertheless, it's a brilliant book because it looks at the pursuit of science almost like a detective story. You're unraveling this mystery and there's a race and all this stuff. So I, in some sense, for Gene Machine, that was a kind of model of making it personal and making it frank and honest, and also honest about yourself. And just as in the Double Helix, they talk about their own mistakes, I talk about all the blunders we made, and also career choices that didn't quite work out, and so on. So because if you're not honest about yourself, you can't be honest about others. So it wasn't so hard. Now, with my second book, it was harder because it was not talking about myself. It was talking about the field of aging. But there, what I did was I tried to personalize it by bringing in characters that I thought were interesting, who had contributed some big advance to the field, and fleshing them out as personalities, just to give a human touch to the process.
Do we have more questions?
Thank you. I've just been reading a book called How Life Works by Philip Ball, and one point he makes is that the link between the gene and the organism's characteristics is now thought to be much more diffuse than it earlier was, and it's a much more... So I was wondering if you could shed some light on that argument.
I don't quite agree with his... I think his views are too extreme. And there is a fashion now... There's an ideological reason for not liking genetics, because if we have different genes, then we're not exactly equal in various characteristics, and that doesn't sit well with very progressive types. And so they would like to minimize the importance of genetics. But the reality is, yes, there's epigenetics, which is environmentally coded, well, environmentally evolved, let's say. But there's, and also the environment does a huge amount to affect various things like your behavior, your perception, and your abilities as well. But biologists have never said that it's all genes and it's a blueprint. They've always said that what actually happens. So Richard Dawkins, for example, said genes are not a blueprint. In a blueprint, you have a fixed formula like in a factory, and every product will be identical. Based on the blueprint, it'll come out exactly as you expect. He said it's much more like a recipe. Different people following the same recipe or under different conditions with different components, ingredients, will come up with something different. And so I think biologists have always felt biology is a combination of genes and environment. But to minimize either one, I think is a mistake. I don't know. How do you feel about that?
Well, I run very contrary to his views, because I think that he does have, as you say, an ideological objection to genetic determinism.
Yeah.
And I think that sometimes people who come from a particularly strong ideological standpoint will interpret everything that you say through a prism of that standpoint.
Yeah.
Good evening, sir. Now you are calling, you have termed this as a gene machine. And of course, this DNA is very, very complex, irreducibly complex. I want your conviction, a sincere view, whether natural selection has actually engineered this DNA, or of course, Earth was here only for around 5 billion years.
Yeah.
But whether it is possible for natural selection to engineer such a very complex machinery,
Yeah,
as you call it.
I think, first of all, biologists would object to the term engineer because that implies a purpose or design. What natural selection simply does is select for traits that survive, okay? And the thing that you propose has been a conundrum ever since evolutionary ideas began. And Darwin really talked about it in the evolution of the eye, because the eye is highly complex, and you can't imagine different parts of the eye existing in isolation. So how would the eye evolve? And he talks about how even a very complicated thing like the eye could evolve from cells that initially were just light sensitive and then eventually added lens and so on. So the reason that it's complex is because it evolves gradually. It's not as if the whole thing is assembled in one go and selected for.
So I work in the field of science, scientific research, but many times many of my answers come from karma philosophy and Advaita. So I have a question which mixes both of that. Pardon my ignorance and then mixing of that. So we say that, okay, when a kid is conceived, within 6 to 7 weeks, you have a basic neural network and a memory to start with. And science says that it is a genetic memory, but in karma says that it is karmic memory. So genetic memory takes all your strains from your ancestors and then pass it on. Karma takes it from your previous lives and pass it on. In your work, have you observed a correlation between these two philosophies?
I think that, to be perfectly honest, the karma aspect of this is not really science, because it's not something you can do experiments and observe. No, I don't mean that in a derogatory way. I mean that science is a particular thing. It's about something you can predict, make measurements, test and observe, and then form hypotheses to refine your ideas, then test those and so on. The other thing there is no experiment you can do to test those ideas. They're a matter of faith or belief, and so they don't fall in the realm of science. You can have them, that's fine. There are many scientists who are religious, and including, by the way, the guy who headed up the Human Genome Project in NIH, Francis Collins, was a devout Christian. So, but you have to somehow compartmentalize the two. One is about your personal philosophy and your life and and how it guides you in life, and the other is about empirical observation and evidence. So evidence is really what science is about.
I'm glad you didn't pass that question to me.
Sorry?
I'm glad you didn't pass that question to me. That would have been very unpleasant. We've got some more questions. Yeah, over here.
Hello. So you have shared your experience when you got the Nobel Prize. What I would like to know what was your feelings when you revealed the structure of ribosome? Did that open a way to more questions?
Oh, definitely. So first of all, I should say the day that we saw that the problem had cracked and we could see parts of the ribosome, in a way, I remember that much more than the day of the Nobel Prize. I mean, they were both big deal, but actually the moment of discovery was really, in many ways, more exciting. But yes, the structure of the ribosome opened up a lot. In fact, we started to understand how the ribosome reads the genetic code, how it stitches together proteins, how antibiotics act on the ribosome to prevent it from working, and we're now trying to understand how the ribosome is regulated, how viruses will hijack the ribosome to make viral proteins and shut down your own proteins. So there's lots that we're learning, and all of it is facilitated. I wouldn't say the structure alone can tell you all this, but the structure really helps you ask more detailed and more sophisticated questions.
I think we've got time for perhaps one more question, or maybe.
I have a question for you. What do you think are the 3 most important questions that you would like to be answered?
You mean in science or in biology or?
In science.
In science, well, I guess, I'm fascinated by neuroscience and the details of how we remember and how we act on memory, how decisions are made. That's, to me, has a question at many different levels, from the molecular all the way to psychology. And so that, to me, is a big question. Another is the origin of life, how life began from very early chemicals.
Thank you very much. I think, have we got time for one more question? Yeah, one more question. Go ahead.
It's a privilege to talk to you. I just have a very simple, the answer is kind of obvious. How does it feel to be one of the only 9 Indians to have won the Nobel Prize? Only 9 Indians have won Nobel Prize in the last 1900 to 2020, 125 years.
Okay, I have to say though, there is, I know in India there is that feeling, but that's only because India doesn't have yet a lot of Nobel laureates. But when an American wins a Nobel Prize, people don't say, "Oh, an American has won." They think more like, "Oh, a guy from Harvard who worked on this or that won." Okay, so the Nobel Prize is not given it's not like cricket where you're playing for India. Okay, it's not about a nationality. It's about doing something significant in science. So you have to think of it that way. I'm very comfortable and indeed enjoy the fact that I grew up in India and came from here. But I don't think the Nobel Prize is I don't think of it as an Indian Nobel Prize. First of all, the work wasn't done here. Even the concepts, I didn't know what a ribosome was when I left India, right? So I don't think of it as an Indian Nobel Prize. But I think Nobel Prizes are not national. They're simply about individuals who did some important piece of work. And that's how you should look at it. And what I would tell Indians is, every year there are Nobel Prizes. You should be interested in all of those as well, because each one of those represents some really important finding, important advance in our understanding of physics or chemistry or biology. And they should be interesting to you regardless of what the nationality is.
Thank you. And this is Aksharam, powered by DC Books. And now a book release is about to take place. We are thrilled to invite Professor Leschziner to release Venki Ramakrishnan's book, Gene Mission. Please, sir.
I have to say, I only saw this book yesterday. Ravi DC kindly offered to publish this book a couple of years ago, and I only saw it yesterday. It's only recently come out. And I don't know Malayalam, so I can't tell you about the translation, but I can say the quality of the printing and the paper and the production and the figures is really fantastic. So those of you who read Malayalam, can go off and buy this book. Thank you.